when you have this guy who is going to have a baby with that woman, and she has 46 chromosomes, and he has 46 chromosomes, and they want their baby to have 46 chromosomes, they need to go through the process of meiosis in order for that to happen. Because if her egg had 46 and his sperm had 46, when that sperm fertilized that egg, you would have 92. And that can't happen. That's like Teenage Mutant Ninja Turtle stuff, or mutants, that's like X-Men kind of thing going on. No, we needed like I had something to do with it. Yes, we needed a process. No, nature needed a process to take this number and divide it in half, divided by 2. And that process is meiosis. So these sex cells have gone through meiosis and therefore only have 23 chromosomes in them without losing any information at all. It's amazing. So when the sperm fertilizes that egg, baby has 46. Ta-da! You're welcome. Hello, my amazing biology students, and welcome to my version of what meiosis is and why you go through it. Okay? So, first of all, the whole reason that animals exist, living things exist, the driving force of all living things is to get their genes into the next generation. That's what drives all living things, okay? And meiosis is how that happens, because meiosis is the process that creates sex cells. All right, so now, I think I say all right a lot. Okay, so right here, I have made for you some DNA. DNA is made up of genes. Depending on the species, there's a couple genes, thousands of genes, tens of thousands of genes. But I'm just going to make believe that this organism only has 7 genes. Genes are specific sequences of DNA that live, genes are specific segments of bases on DNA. That's where genes are. They're on your DNA, or on any organism's DNA. Not just yours, also the banana, also the cat, and the bird, and the tree, and the cricket. You get the point. Then the shark. Anyway, so, but you have to realize that organisms don't just have one parent's DNA, they have both parents' DNA, right? All organisms have 2 pairs, 2 variations of the same genes, because they got one from one parent and one from the other parent. These variations, we call them alleles. I'm going to flip this over. So these are the variations that I made up. So let's say gene 1 is eye color. And this parent had brown eyes, and this parent has the alleles for blue eyes. Because, and I'm going to write it down here for you. Okay? All right. Alleles are the variations of genes. You get one from mom and one from dad. whether you're a parrot or a something that lives in the ocean, a dolphin, or a squid. Sure. Okay. So, gene 1, this parent had brown eyes, gene for brown eye, or the alleles for brown eyes. This parent had the alleles for blue eyes. This parent has this allele or these alleles for wavy hair. This parent had straight hair. This parent had dimples. Oh, so did that one. Okay. Attached earlobes. We have alleles for attached earlobes with that parent. This parent had free earlobes. Blonde hair. This parent had brown hair. This parent was left-handed. This parent was right-handed. This parent had no widow's peak. This parent had a widow's peak, which you can't read at all because it's black on gray, but take my word for it. That's what I wrote down there. If you don't know what a widow's peak is, think Dracula. That V that comes into the center of your forehead, that's a widow's peak. Anywho, so genes come in variations. Those variations are called alleles. And we are going to start talking about alleles pretty heavy when we talk about meiosis and we talk about Mendel's Punnett squares and we do all those funky probabilities that are so amazing and we all love. Okay, now how do we get this information into the offspring without losing any of this information? I'll tell you how. I knew you were wondering, and I'm going to tell you because I know it's been a burning, burning question all of your lives. And now you will have the answer to it. Now I'm just playing with Legos. You have learned prior to today about the cell cycle, right? The cell cycle. During the cell cycle, DNA gets duplicated. Look at, oh, I did it kind of backwards, didn't I? I have gene 1. But you can see gene 1 represents brown eyes. That's kind of a cool way to do it. During interphase of the cell cycle, your DNA gets duplicated. So this parent's DNA was duplicated and this parent's DNA was duplicated. Okay? That's an important step. That's how we can create sex cells during meiosis without losing any information. Es muy importante, right? All right, so we've gone through interphase. We have duplicated the DNA, and that DNA has now coiled into these beautiful X-shaped things called, oh, what's going on here? Let's do this. There we go, that looks better. Coiled into these beautiful X-shaped structures called chromosomes. These are called chromosomes. The reason your DNA coils into chromosomes, is because your DNA is a very long molecule. If you took it out of the nucleus of a cell and made it straight, it would be about the length of a car. That's a very long molecule with a lot of information on it that is tucked and folded and twisted and stuffed into the nucleus of a cell. So in order to make sure that all of the information gets transferred, it coils into this chromosome shape. So the information doesn't get forgotten. Some mistakes still happen to get made, but fewer than if the DNA was not in a chromosome shape. So this X shape of coiled DNA is called a chromosome. All right? Now, you'll notice that I said this was dad's, right? This parent's chromosome has the same information as that parent's chromosome, right? We're talking eye color for gene 1. We're talking hair, wavy or straight for gene 2. So, you know what this means? These two chromosomes are called homologous. Homo means same. Homo means same. Homologous. They are homologous chromosomes. Chromosomes. Okay? Because they carry the same information. Now, let's go into the first phase of meiosis. Oh, I have to give you some, I'm sorry, I have to give you some more information. Also, these chromosomes, they are homologous. I've already said that. They are consisting of chromatids. Each chromosome consists of a sister chromatid. Let me get my arm around there. I can't do it. I can't do it. Chromatid. Okay? So there's one sister chromatid and two sister chromatid. Sister chromatid. All right, so each chromosome is two sister chromatids. So that means, how many sister chromatids are there? 1, 2, a 3, a 4. There are 4 chromatids. Those make up what's called a tetrad, because tetra means 4. Okay? So that's what's going on right here. Now, we're going to put this tetrad to the test. I don't have a thing to wipe off the dry erase marker, so I'm using my hands. Sorry. Okay. So, meiosis, let's go. The first thing we're going to do is we're going to go into prophase 1, because something immensely important happens during prophase 1. We call it 1 because meiosis has 2 stages. So it will go through prophase, metaphase, anaphase, and telophase 1, and then it goes into stage 2, prophase 2, metaphase 2, anaphase 2. You get the point. So you have to always designate 1 or 2. We are in prophase 1, the first stage. Okay, so these homologous chromosomes, and if you could see a picture of it, it's amazing. They are wrapped around each other. I mean, they're like, we share so much of the same information. I could just share with you and exchange. Let's exchange information. And that is exactly what they do, by the way. They wrap around each other and exchange genetic information. And I'm going to do that right now, so you will see what the result is. Okay, so now, once this magical event of crossing over occurs, this is a huge, important event. You have to understand this. You have to know what this is. Crossing over is how we get genetic variation within offspring. It's why when you look at offspring, they're not genetically identical to each other, because this event took place, creating completely never before seen chromosomes. These are all unique variations of chromosomes as a result of that crossing over. Mimi, I love you. Let's exchange genetic information. It's time to cross over. Okay? Now, I want you to keep in mind, and we're going to learn about this later, but I'm just going to drop this on you right now while you're looking at it. If you get two alleles that are very close together on a chromosome, then sometimes they move as one. Because they're so close, they're never really separated, ever. They will both move to another chromosome during crossing over. But if you have alleles that have a nice amount of separation between them, they'll move independently. We'll talk about that in a minute. So you have to know this. I'm going to use a different color to really. Now I use blue. You have to know that crossing over happens during prophase 1, and it creates all of this genetic variation. It's creating completely unique chromosomes. Okay? All right, now let's go on to the next phase, which is metaphase. So during metaphase 1, the chromosomes line up in the middle of the cell, okay? But the second most popular, yes, everyone's talking about it. It's the most popular. No, the second most important event occurs. And this is called, if I can fit it in here, let's see, independent. Oops, let's spell it correctly. Independent assortment. And so what that means is the cell doesn't care which chromosome goes to which pole. It doesn't matter. And in a real organism, by the way, you can have 6 or 7 or 10 chromosomes. So some go on this side, some go on that side. It's random. Every time it happens, it's random. So that random assortment into the next cells is another way of creating genetic variation in the sex cells that will be created from this process. So independent assortment also, along with crossing over, let me get my blue marker. Oh, you know it's important. Oh, there we go. Also an important thing to know that happens during metaphase 1. Okay, now, let me kick it up a notch, just for a second sec, okay? You will find, depending on where the traits are on the chromosome, that if they are very close together, you know, like maybe these two or these two or these two, they will move like one. They are called linked, and they do not really separate because they're so close. So during that crossing over part, they're just always together. So even though they're two traits, those two traits are linked because of their closeness. So you always see them together in the offspring. Now, if the traits are far apart, then oftentimes one goes there, one goes there. They are not linked, and you will see different traits, blue eyes and widow's peak, or brown eyes and no widow's peak. You know, they're not locked into another chromosome, so to speak. Okay, so where these traits are on the chromosome also plays a role in the outcome, and we'll see that when we do Punnett squares. and it is during anaphase that they get pulled apart and the cell elongates. I'm gonna elongate this cell right now. Elongated cell, right? That's anaphase 1. After anaphase, you go into telophase 1, where the cell is going to begin to separate. Telophase 1, cytokinesis. I should say telophase and cytokinesis. I'm a horrible speller. Kinesis, whatever. That's, there's no connection between spelling and intelligence. So don't let anyone tell you that. Horrible speller. I heard Einstein was a horrible speller too. Not that I'm like Einstein, but probably close. No, I'm just joking. Okay, so at this point, we now have 2 cells. But what have you noticed about these cells? In our beginning cell, we had 2 chromosomes. Each of these cells have how many? That's right. Each of these cells has only 1 chromosome. But did we lose any information? When we have all the information, this was the information that just got mixed up and exchanged, right? But this is all the information. So during that first stage of meiosis, we have already reduced the chromosome number by half without losing any information at all. Amazing. It is an amazing feat. All right, now let's go on to stage 2. We already have our chromosomes. They've already crossed over. That happened during prophase. Prophase. We don't have to go through it again. So we're gonna hop, skippity hop into metaphase 2, okay? Now, the dealio with metaphase 2 is that we have 2 cells going through it. So we have centrioles. So metaphase, they have lined up, in the middle of the cells. And we're going to go into anaphase where these chromosomes are pulled to each side of the cell. All right, here we go. Anaphase 2, we have the chromosomes being pulled to each end of the cell. That is always what's happening during anaphase. The chromosomes are always being pulled to the poles of the cell. Notice how they're apart now. But still, have we lost any information? No. Each cell will still have all the information they need. Amazing. Okay, next phase after anaphase, do we remember what it is called? It's called telophase. Have 4 sex cells made. These sex cells are also called gametes. Do you have any example of gamete? Like, what is a female gamete called? It is called an egg. What is a male gamete called? It is called a sperm. A sperm. Every time you eat an egg, you're eating a chicken's gamete. It is the largest cell in the female body. So think of an ostrich egg. It's huge, but it's 1 cell. An ostrich egg is 1 cell. A chicken egg is 1 cell. So it's amazing. When you have eggs, you are having a gamete. Sperm cell, also. Sperm cell much smaller than an egg, but the largest cell in the male body. Pretty cool stuff. So now at the end of meiosis, we've reduced the chromosome number in half, and we have created 4 gametes, or sex cells. And that is how species are able to go from their full amount of chromosomes to half the amount of chromosomes without losing any information. And it's just an amazing process. So let's just recap. You need to know the 2 events that create variation. And they are, okay, Miss Passerelli, of course I know what they are. They are, are you already saying them to yourself? Hold on a second, let me write them down. We have, ooh, that's a very new crossing over. And independent assortment. Right? This happens during prophase 1. An independent assortment happens during metaphase 1. Oh, you can't. You didn't really see the metaphase 1 part, did you? Let me. Twing. That looks like a 2. No. Metaphase 1. There you go. All right. Also, what is created at the end of meiosis? I'm just giving you the kind of questions that I would ask my students. What is created at the end of meiosis? 4 gametes, right? Gametes are also known as sex cells. Are they identical, or are they genetically what? Identical or unique? They are unique. That is correct. They are genetically unique. Awesome sauce. So,